Hello everyone, I am Radhika, Assistant Professor from Department of Biochemistry, SBRR Mahajana First Grade College. Today I will be discussing about Meselson-Stahl experiment, which serves as an experimental proof for semi-conservative mode of DNA replication. Before starting with the actual topic, let's have a brief introduction about molecular biology, DNA replication and semi-conservative mode of replication. So as we all know, molecular biology is a well established branch of biology and biochemistry which specifically concerned with the study of different life processes at its molecular level. Molecular biology includes study of different biomolecules like carbohydrates, proteins, nucleic acids, lipids, their composition, their structure and functions of these biomolecules in different life processes. Molecular biology is one such established branch which lays a strong foundation on the fundamental aspects of molecular mechanisms like replication, transcription, translation and other cellular processes. In simple, we can also say that the gist of molecular biology is the central dogma. Central dogma basically it's a fundamental framework to understanding the transfer of information sequentially between the biopolymers. So, central dogma is a sequential transfer of information which includes the mechanisms like replication, transcription, translation and also reverse transcription mechanisms. In molecular biology, different discoveries has been made and molecular biology is being laid with a number of milestones. Elucidation of DNA replication mechanism is one of the major milestone in the field of molecular biology. DNA replication is a phenomenon in which the parent DNA will produce an exact copy of itself. That is, in simple we can say that DNA replication is a mechanism where the two daughter DNAs will be produced from a single parent DNA. DNA replication is being explained with three different models. That is, we have three different models for explaining the mechanism of DNA replication: semi-conservative mode of replication, conservative mode of replication and dispersive mode of replication. Today I will be focusing only on semi-conservative mode of replication. So this semi-conservative mode of replication is a model of replication mechanism explained in which the parent strand serves as the template strand for the synthesis of new complementary daughter strands. So in simple, we can say that semi-conservative mode of replication is a phenomenon or a mechanism wherein the parent strands will get separated. How does the parent strand separates? The hydrogen bond between the nucleotides that is holding the two strands of DNA together will be broken down. With the breaking of the hydrogen bonds between the nucleotides, which leads to separation of the two parent strands, the separated two parent strands will now serve as the template strand for the synthesis of new complementary daughter strands. This is what the semi-conservative mode of replication is. And the daughter DNAs will retain one of the parental strands, so that the daughter DNAs contain, out of two strands, the daughter DNA contain one parent strand and one newly synthesized strand. This is semi-conservative mode of replication. And this hypothesis was proposed by two eminent scientists, Francis and Crick, and the hypothesis was proposed based on the model of DNA structure. And it is known that any of the hypothesis that is being proposed will not be accepted until and unless an experimental evidence is given supporting that hypothesis. One such attempt to give experimental evidence was Meselson and Stahl experiment. Meselson and Stahl experiment serves as a evidential proof for semi-conservative mode of DNA replication. So, we can say that Meselson and Stahl experiment gave the experimental evidence of DNA replication to be semi-conservative type. It was introduced by Matthew Meselson and Franklin Stahl in 1958. Two of these scientists, they designed a set of experiment wherein they selected particularly an E. coli organism as the model organism. E. coli only was taken as a model organism because of number of advantages it had within the E. coli organism. So, Meselson and Stahl used E. coli as model organism to explain the semi-conservative mode of replication. So, the advantage in E. coli cell is that E. coli cells, they have a very short duration of time. E. coli cells can be easily cultured in a culture medium under laboratory conditions, and they have a very short lifespan, that is each E. coli cell can replicate and produce one generation of cells in a span of 20 minutes. So, at every 20 minutes one generation of cells would be produced, so that the E. coli cells is completing one replication cycle for every 20 minutes. So, that is the reason why E. coli cells was taken as a model organism. And the experiment was designed in such a way that two of the nitrogen isotopes were selected, were considered for the experiment. N15 nitrogen, which constitutes heavy isotopic form of nitrogen, and N14 nitrogen, which constitutes light isotopic form of nitrogen. The advantage of these two nitrogen forms is that when the nitrogen is being subjected for density gradient centrifugation, heavy nitrogen, that is N15, would appear as a band at the bottom of the test tube, and N14 which appear as a band at the top of the test tube, and the hybrid of which will appear at the center of the test tube as a hybrid molecule. So, nitrogen isotopes were selected, and initially, starting with the experiment, set of E. coli cells were grown on a medium containing only N15 isotopic form of nitrogen for many generations, and this is to ensure that almost all the cells, E. coli cells, will incorporate the N15 heavy isotopic form of nitrogen into the genome of the E. coli cells. After which the set of E. coli cells were grown on a medium containing N14, that is light isotopic form of nitrogen, for many generations. After growing these cells, at the end of every generation, that is once the cells complete one cycle of replication, the cells were isolated from the culture medium and were subjected for density gradient centrifugation in cesium chloride density gradient medium to check the density of the DNA molecule in the bacterial cells. The whole experiment was designed in this way, and the experiment was carried out in such a way that firstly the cells were grown in N15 containing medium for many generations. After ensuring that all the cells have incorporated N15 heavy isotopic form of nitrogen, the cells were then grown in N14 containing medium. At the end of every one generation, that is after the cells have completed one cycle of replication, one round of replication, the cells were isolated from the medium and subjected for density gradient centrifugation. The results observed were in such a way that the cells which were grown in only N15 containing culture medium and subjected to density gradient centrifugation. The DNA of the bacterial cells, after centrifugation, they formed band at the bottom of the test tube. This band which is formed at the bottom of the test tube is indicating that the DNA of the bacterial cells contain only N15 heavy isotopic form of nitrogen. After the cells were being transferred to medium containing N14, that is light isotopic form of nitrogen, after completing one round of replication, again the cells at an interval time of 20 minutes, as 20 minutes is considered to be a completion time for one life cycle. At the end of 20 minutes, so at a regular interval time of every 20 minutes, at the end of 20 minutes, first generation cells were isolated and subjected for density gradient centrifugation. The result obtained was that the band was formed at the center of the test tube. Single band was formed at the center of the test tube. This single band at the center of the test tube suggested that the DNA of the bacterial cells contained hybrid DNA, that is the DNA which contains both N15 and N14 isotopic forms of nitrogen. So, which can be said that N15 isotopic form of nitrogen is the parental strand and N14 isotopic form of nitrogen is present in the daughter strand. So, therefore, the bacterial cells contain DNA as the hybrid DNA containing both N14 and N15 isotopic forms of nitrogen. Next, after completing the second generation, that is second round of replication, after completing 40 minutes of time, at the end of 40 minutes, ensuring that the second round of replication is being completed, the cells were again isolated from the medium and subjected for density gradient centrifugation. The result observed was that the bacterial cells, the bacterial DNA, it formed two bands, one at the center of the test tube and one at the top of the test tube. This suggested that the band which is formed at the center of the test tube contained hybrid DNA which contained both N14 and N15 isotopic forms of nitrogen, and the band which was formed at the top of the test tube, it included only N14, that is light isotopic form of nitrogen. And further, the third generation was continued after completing the third round of replication. At the end of 60 minutes, the cells were again isolated and subjected for density gradient centrifugation, and the result observed was again two bands was formed, one at the center and one at the top of the test tube, indicating or suggesting that the center band is because of the DNA containing hybrid N14 and N15 isotopic forms of nitrogen, and the band at the top of the test tube is because of N14 isotopic form of nitrogen containing DNA. So, like this, the experiment was continued for many generations, and as the experiment continued with the same N14 medium, as the cells were continued to be grown in the same N14 medium, and after successive generations, number of successive generations, the result observed was in such a way that there was always two band appeared, one at the center and one at the top of the test tube. But gradually the concentration of the band which was formed at the center of the test tube gradually decreased. The concentration of the band formed at the center of the test tube gradually decreased, and the concentration of the band which was formed at the top of the test tube gradually increased. This increase in concentration with the top band, the band which is formed at the top of the test tube, is because the cells are being grown successively in a medium containing only N14, light isotopic form of nitrogen. Because of this continual growth of bacterial cells in a medium containing only N14 isotopic form of nitrogen, the concentration of the bacterial DNA containing N14 isotopic form of nitrogen increased, and the concentration of the DNA containing hybrid DNA, that is containing both N14 and N15 isotopic forms of DNA, gradually decreased. So, after many successive generations, after completing a number of many successive generations of growth in N14 containing culture medium, the density gradient centrifugation result and observation was that the bands formed at the center of the test tube gradually decreased, the concentration of the band at the center of the test tube gradually decreased, and the concentration of the band at the top of the test tube gradually increased. So, this was the entire experiment which was done, and end of the experiment the result was very much clear that initially when the cells are grown in N15 medium, the cells incorporated only N15 isotopic form of nitrogen, so that it produced a single band at the bottom of the test tube. Then when the cells were transferred from N15 to N14 containing medium, N15 light isotopic form of nitrogen containing medium, the cell started incorporating N14 light isotopic form of nitrogen into the genetic material. So, the band was formed at the center of the test tube, indicating it to be a hybrid DNA band. And with the successive generations, further continuation with the experiment and successive generations of successive completion of generations of cycles, at the end of every generation bands appeared as two bands, one at the center and one at the top, indicating that there is both hybrid DNA containing both N14 and N15, and the newly formed daughter strands incorporated, the newly synthesized daughter DNA incorporated only N14 isotopic form of nitrogen, which results in band formation at the top of the test tube. So, with this the experimental result was concluded, saying that based on the observation and the experimental results, Meselson and Stahl concluded that the DNA replication mechanism is semi-conservative mode of replication. So, with this experimental proof, it is proved that the hypothesis given by Francis and Crick, saying that DNA replication mechanism is a semi-conservative mode of replication, was proved with experimental evidence given by Meselson and Stahl. Investigation of semi-conservative mode of replication did not halt at this point. It was further continued by different number of scientists by performing different sets of experiments, some of which we can quote like experiment done by Hershey and Chase using bacteriophage as the model organism, and even this proved DNA as the genetic material, and other further experiments was by Taylor and colleagues, who also investigated on semi-conservative mode of replication and who also worked to provide experimental evidence for semi-conservative mode of DNA replication mechanism. So, with this I am completing with the discussion on Meselson and Stahl experiment, that is an experimental proof for semi-conservative mode of replication. Thank you one and all.